Welcome back to the deep dive. Today we are tackling a beast. And I don't really mean that metaphorically.
No, this one is literal.
We're diving into the single largest, most biologically diverse, and frankly, clinically dominant family of bacteria you are ever going to encounter in your medical career.
It is the absolute heavyweight champion of the microbiology lab.
So if you're a medical student, a resident, anyone in healthcare, this is the one where the rubber really meets the road.
We are talking about the Enterobacteriaceae.
The Enterobacteriaceae. It's a mouthful. And when you look at the source material, we're working primarily from Chapter 25 of Murray's, with a little help from Jowett's. It's honestly overwhelming.
It is. It's a huge list of names.
Dozens of genera, hundreds of species. It feels like trying to memorize a phone book where everybody has a similar last name.
And that's the trap. If you try to memorize it like a phone book, just a list of names and bugs, you will fail.
Or at least be miserable doing it.
Exactly. The trick, and what Murray really emphasizes, is to understand them not as a list, but as enteric architects.
Enteric architects. I like that image. It implies they're building something.
They are. These bacteria essentially build the environment of our gut. They're the most common gram-negative rods in your gastrointestinal tract. They help structure the microbiome.
But there's always a but.
There is. They're also architects of destruction. It's this complete duality. They can build a peaceful community in your colon, but if they get into the wrong place or pick up the wrong gene,
they can tear you down.
They can tear down your host defenses, cause systemic shock, dissolve your kidneys. It's a huge spectrum.
So our mission today is to decode that duality. We're going to build a structural, functional, mental model. We need to understand why they do what they do.
Right. So we'll start with the family blueprint, what makes them all the same.
Then we'll hit the prototype, Escherichia coli.
And then the specialized invaders, Salmonella, Shigella, and Yersinia.
And finally, the opportunists, the hospital gang like Klebsiella and Proteus. And we'll wrap it all up with some rapid-fire practice questions to lock it in.
Sounds like a plan. Let's go deep.
Perfect. So let's unpack that blueprint. If I handed you a Petri dish and said, is this family? What are you looking for? What's the ID card for the Enterobacteriaceae?
Well, physically, they're pretty unpretentious. Under the microscope, they're just moderate-sized gram-negative rods.
So pink Tic Tacs.
Pink Tic Tacs, exactly. And Dewitz adds a key detail. They're non-spore forming.
Which is important because it separates them from some of the gram-positive nasties like Clostridium or Bacillus.
Right. These guys don't hibernate. They're always active, always ready to go.
Okay, but you can't tell them apart just by looking. A gram-negative rod looks like a gram-negative rod.
Morphology gets you in the door, but biochemistry gets you a seat at the table.
Ah, so we need the lab tests.
You need the big three biochemical rules.
Yeah.
This is the triad for your exams and for the lab. Rule number 1, they all ferment glucose. Every single one.
Okay, that makes sense for a gut bacteria, right? Lots of sugar down there.
Precisely. They're metabolic engines. Rule number 2, they reduce nitrate to nitrite.
And this is about how they breathe without oxygen.
In a way, yeah. They can use nitrate as a terminal electron acceptor when oxygen isn't around.
Yeah.
It's part of why they're so good at surviving in the gut.
So they can breathe nitrate.
You could say that, but here's where it gets really interesting. Rule number 3, the deal breaker.
This is the one that separates them from the imposters?
Correct. They are cytochrome oxidase-negative.
Okay, why is that the deal breaker? Why do we care so much about this one enzyme?
Because there is another major clinically devastating group of gram-negative rods that causes a lot of the same infections. Pneumonia, sepsis, UTIs.
Pseudomonas.
Exactly. And Pseudomonas is oxidase positive.
So in the lab, you run that quick oxidase test, and if the little paper turns blue?
You're dealing with Pseudomonas or maybe Vibrio. If it stays colorless, it's oxidase negative.
Yep.
And you are squarely in the territory of the Enterobacteriaceae.
So the oxidase test is the big fork in the road.
It is. Think of them as the standard army issue bacteria. They're rugged. They're facultative anaerobes.
Meaning they can live with or without oxygen?
They're not picky. They can switch gears, use aerobic respiration or fermentation. They are built to survive almost anywhere.
Okay, so once we know we're in the family, we have to start telling them apart. And Murray makes a huge deal about McConkey agar. I feel like McConkey is the sorting hat of microbiology.
It really is the sorting hat. That's a perfect analogy.
So what does it do?
McConkey agar is both selective and differential. It does two things. First, it has bile salts and crystal violet.
Which kills off the gram-positives.
Right. It inhibits their growth. So if something grows on McConkey, you already know it's a tough gram-negative gut bug that can handle bile.
Okay, so it clears the field, but how does it sort the family?
It sorts them based on one simple question. Can you ferment lactose?
This is the pink versus colorless divide.
Yes. The agar has lactose in it and a pH indicator called neutral red. If the
Mhm.
bacteria ferment the lactose, they produce acid,
And the acid drops the pH.
which turns the neutral red indicator a bright, hot pink,
Hmm.
or sometimes purple. If they don't ferment lactose, they eat the peptones in the agar instead. And that
It's
produces...
ammonia, which is basic.
Mhm.
So the colonies just look colorless or kind of beige.
So it's a visual binary. Pink or not pink. So who are the pink ones? Who are the lactose fermenters?
The mnemonic I use is CEEK.
Cake.
Citrobacter, Escherichia, that's E. coli, Enterobacter, and Klebsiella. If you see hot pink colonies on that plate, it's probably one of those.
These are generally your opportunists, right?
Yeah.
The good guys got bad.
For the most part, yes. Though Dovets does add a little curveball with Serratia.
Right. What about Serratia?
It can be a slow fermenter, so it might take a couple of days to turn pink. It can trick you at first.
But CEK are your rapid fermenters, and then you have the colorless ones, the non-fermenters.
And this is a vital clinical decision point. The primary pathogens, the ones that are almost never normal flora and almost always mean trouble, they do not ferment lactose.
You're talking about
Shigella, Salmonella, and Yersinia. And also Proteus, which is more of an opportunist, but it's also colorless. So if you have a patient with bloody diarrhea and the lab calls and says, we've got growth on McConkey and it is a lactose non-fermenter, your alarm bells should be screaming.
Absolutely. You've just narrowed it down to the real bad actors. You've ruled out a typical E. coli or Klebsiella. You're hunting for Salmonella or Shigella.
Okay, let's talk about their armor. Murray describes this antigenic structure that sounds like a spacesuit. You've got O, H, and K antigens. And I think students just memorize the letters.
That's a common pitfall. This isn't just for exams. This is serology. It's how we track outbreaks in real time, and they are physical structures.
So let's start with O, the O antigen.
O stands for Ohne-Hauk, which is German for without breath. It refers to the somatic antigen.
Somatic meaning body.
Right. The O antigen is the outermost part of the lipopolysaccharide, or LPS, in the cell wall. It's a long chain of sugars that sticks out from the bacterium like hair.
And it's really variable.
Hundreds of types. It's a sugar chain. So you can swap out the sugars, change the linkages. This is why we have E. coli O157 or O111. It's identifying the specific sugar pattern on the skin of the bacteria.
So the O is the body of the suit. What about the K?
The K antigen stands for capsule, German for capsule.
Ah, the capsule.
This is a polysaccharide layer that covers the O antigen. It's an outer shell, a force field.
What does the force field do?
It protects the bacteria from phagocytosis. If a macrophage tries to grab it, the capsule is slippery, or it hides the antigens the macrophage would recognize. It's a stealth cloak.
And sometimes it gets a special name, right? Like the Vi antigen.
Exactly. In Salmonella typhi, the capsule is called the Vi antigen for virulence, but structurally it's just a K antigen.
Okay, and finally the H antigen.
H is for Hauch, breath or mist. This refers to the flagella,
The tail.
the protein that makes up the whip-like tail. So if a bacterium has an H antigen, it's motile. It can swim.
Do they all have them?
Nope. There are always exceptions in microbiology. Shigella and Klebsiella are generally non-motile. They have no flagella.
So no H antigen.
No H antigen. And that's a great way to spot them in the lab. If you have a bug that's just stuck in one spot on the agar, it might be one of those two.
Okay, before we dive into the individual bugs, we have to talk about the weapons. Murray lists a common arsenal they all share, and the big one, the nuclear option, is endotoxin.
Lipid A.
Lipid A.
It's the universal weapon. So we said the O antigen is the outer hair of the LPS. Well, lipid A is the anchor. It's the part that's buried in the bacterial membrane.
And it stays buried as long as the bacterium is alive.
Mostly, yes. But when the bacteria die or get lysed, or even when they're dividing rapidly, this lipid A gets released into the host's body. And our immune system absolutely freaks out.
This is what causes gram-negative shock.
This is the cause of septic shock. Lipid A binds to a receptor on our immune cells called toll-like receptor 4, or TLR4.
And that sets off a firestorm.
A cytokine storm. Massive systemic release of IL-1, IL-6, TNF-alpha. It activates complement. It activates the coagulation cascade.
So you get fever, your blood vessels get leaky, your blood pressure tanks.
And you get disseminated intravascular coagulation, or DIC. You're clotting and bleeding at the same time. It's a total systemic meltdown.
And it doesn't matter if it's E. coli or Salmonella or Yersinia.
If you have enough lipid A in your blood, you go into shock. It's a clumsy weapon, but it's devastatingly effective at causing chaos.
But they also have something more sophisticated. Murray describes the type 3 secretion system. He calls it a molecular syringe.
This is one of the coolest things in all of microbiology. It is fascinating evolutionary engineering. If lipid A is a bomb, the type 3 secretion system is a sniper rifle.
Okay.
It's a complex protein structure. It looks exactly like a hypodermic needle that the bacterium builds on its surface.
And when it contacts a host cell,
It punches a hole right through the host membrane and injects bacterial proteins, they're called effector proteins, directly into the host's cytoplasm.
so it's not just dumping toxins nearby, it's injecting them straight into the control room of the cell.
It's hijacking the cell from the inside. These proteins can rearrange the host cytoskeleton to force the cell to swallow the bacteria. That's how Salmonella gets inside.
Wow.
Or they can kill the immune response from within. They can tell a macrophage, do not eat me, or even commit suicide. Yersinia, Salmonella, Shigella, some E. coli, they all use this. It's essential for their virulence.
Okay, one more weapon. The siderophores. The scavengers.
Right. Iron is the currency of life in the human body. Bacteria need iron to replicate. But our bodies know this.
So we hide it.
We lock our iron away in hemoglobin and ferritin and transferrin. We try to starve them. It's a form of nutritional immunity.
So the bacteria fight back.
They've evolved these molecules called siderophores, like Enterobacter. They're high-affinity iron-chelating molecules.
They're like little iron magnets.
The bacteria send them out like little thief drones. They have a higher affinity for iron than our own proteins do. They rip the iron right off our transferrin and bring it back to the bacteria.
It's metabolic theft.
That's a perfect term for it.
Okay, so we have the blueprint, gram-negative rods, glucose-positive, oxidase-negative, nitrate-positive. We've got the McConkey sorting hat. We've got the spacesuit antigens and the molecular syringe. Now let's meet the family.
Let's start with the prototype.
The one, the only, Escherichia coli.
E. coli, the Jekyll and Hyde of the gut.
That's the perfect way to describe it because on one hand, it's the most abundant aerobe in my colon right now, just minding its own business.
It produces vitamin K for us. It's a good neighbor.
But on the other hand, it can become an absolute monster.
And that's the key concept. A harmless lab strain of E. coli becomes a killer by picking up a plasmid or getting infected by a bacteriophage that's carrying a toxin gene. It scavenges genetic material.
So Murray breaks down the gastroenteritis strains into this alphabet soup.
Yes.
E-tech, E-pec, E-A-E, stick, E-O-E.
It is an alphabet soup, and it's a nightmare for students to memorize. So let's break them down, not by acronym, but by mechanism.
Let's make it sticky. First up, E-tech, enterotoxigenic E. coli.
The T is for traveler. This is Montezuma's revenge. You go to a developing country, drink the water, and get explosive watery diarrhea.
And the mechanism here is purely toxin-mediated, right? The bacteria don't actually invade the gut wall.
Correct. They attach to the surface of the small intestine, but they don't dig in. They just sit there and secrete toxins. No inflammation, no tissue destruction, just fluid.
And the mnemonic everyone uses for this is labile like the air, stable like the ground.
Yes. So E-tech produces two toxins, heat labile toxin, or LT, and heat stable toxin, or ST.
Labile means it's destroyed by heat.
And stable means it survives heat. But the mnemonic helps you remember what they do. The labile toxin acts exactly like cholera toxin. It increases AMP.
Air equals can't pee.
Right. And ADP ribosylates a G protein, which permanently turns on adenyl cyclist, leading to high CMP.
Yeah.
This forces the CFTR channel open, dumping chloride into the gut. Water follows chloride.
And you get watery diarrhea.
A flood of it.
And the stable toxin.
Heat stable toxin acts on CGMP. Ground equals CGMP. It binds guanylate cyclist. Same end result, just a different pathway. Both toxins cause the cells to pour water and electrolytes into the intestine.
So E-tech is basically cholera light.
That's a great way to think about it.
Okay, next letter. E-pec, enteropathogenic E. coli.
The P is for pediatrics. This is a major cause of infant diarrhea, especially developing countries. And it's really dangerous because babies can dehydrate so quickly.
The mechanism here is visually terrifying. Murray describes it as attachment and effacement.
It's incredible. The bacterium lands on an intestinal cell. Then it uses that type 3 secretion syringe we talked about to inject its own receptor into the host cell membrane.
Wait, it brings its own docking station.
It brings its own docking station. It injects a protein called TIR, which inserts into the host membrane. Then the bacterium binds tightly to TIR.
Wow.
Once it's attached, it injects other proteins that cause the microvilli to just dissolve. That's the effacement. They melt away.
And then it builds the pedestal.
It rearranges the host's actin cytoskeleton to build a literal pedestal that pushes the bacterium up. So it's sitting on a throne of your own actin.
While destroying your ability to absorb nutrients.
Exactly. That's why it causes malabsorption and chronic diarrhea in kids. The genes for this whole process are on a specific pathogenicity island called the LEE.
The locus of enterocyte effacement.
You got it.
Next up is EAEC, enteroaggregative.
Think stacked bricks. These bacteria clump together on the surface of your intestinal cells in a formation that looks just like stacked bricks.
And they form a biofilm.
A thick biofilm, yes. They produce toxins, and they're associated with chronic diarrhea and growth retardation in children. We also see them a lot in HIV patients.
Okay, now we get to the big one. The one that makes the evening news. STEC. Shiga toxin-producing E. coli.
Also known as EHEC, or enterohemorrhagic E. coli.
This is the villain.
This is the one. The most famous serotype is O157.H7. This is the one you get from undercooked beef, contaminated spinach, petting zoos.
Why is it so much worse? Why does this one kill people?
It all comes down to the Shiga toxin. E. coli stole this gene from Shigella via bacteriophage.
And the toxin is nasty.
It's incredibly potent. It enters the cell and it finds the ribosome, the cell's protein factory. It specifically cleaves the 28S rRNA of the 60S ribosomal subunit.
Which just stops protein synthesis dead.
It shuts down the fat, the cell dies. In the colon, this causes hemorrhagic colitis, massive cell death, ulcers, bloody diarrhea.
But the real danger is when the toxin gets into the bloodstream.
Exactly. The toxin has a specific receptor called GB3. And guess where GB3 receptors are highly concentrated?
In the kidney.
In the endothelial cells lining the glomerulus of the kidney. The toxin binds there and kills those cells. This exposes the basement membrane.
And platelets rush in to patch the holes.
Right, which sounds like a good thing, but it goes haywire. You get massive platelet clumping, which uses up your platelets. That's thrombocytopenia.
Low platelet.
Then, as red blood cells try to squeeze through these clogged, damaged vessels, they get sheared apart. That's microangiopathic hemolytic anemia.
And finally, the kidneys just get so clogged they shut down.
Renal failure. And that is the triad of HUS, hemolytic uremic syndrome, anemia, thrombocytopenia, and renal failure.
And there are two critical things to know about STEC O157.H7. First, in the lab.
Unlike almost all other E. coli, it does not ferment sorbitol.
So labs use a special agar.
Sorbitol macaque agar. Normally, coli is pink. O157H7 is colorless. It's a fantastic screen.
And second, clinically.
And I want to really stress this.
Do not give antibiotics.
Why not?
For a couple of reasons. Certain antibiotics, like fluoroquinolones, can actually induce the phage that carries the toxin gene, causing the bacteria to produce more toxin. Also, killing the bacteria all at once releases a massive bolus of preformed toxin into the system, so you make it worse.
Studies clearly show that giving antibiotics increases the risk of developing HUS. The treatment is supportive care only. It's a vital do-no-harm point.
Okay, quickly. EIEC. Enteroinvasive.
That one's basically Shigella light. It invades the colon, causes some bloody diarrhea, but it's pretty rare compared to the others. It lacks the Shiga toxin, so HUS isn't really a concern.
All right, let's move out of the gut. E. coli is the #1 cause of UTIs.
Yes. Uropathogenic E. coli, or UPEC, and it has special tools for this job. The main one is P-pili.
P-pili?
These are PPR fimbriae that are like grappling hooks. They bind specifically to the P blood group antigens on your bladder and kidney cells.
And this is important because
urine flow is a powerful defense. We're constantly flushing bacteria out. But the P-pili allow E. coli to hold on tight against the flow. And it's what lets them climb the ureter to cause pyelonephritis, a kidney infection.
And lastly, for E. coli, neonatal meningitis.
This is just tragic. It's the second most common cause of meningitis in newborns, right after group B strep.
And the virulence factor here is the capsule.
Specifically, the K1 capsule.
K1, what's so special about it?
The K1 capsule contains sialic acid. And sialic acid is a normal component of human cell membranes, especially on our neurons.
So it's camouflage.
It is molecular mimicry at its finest. The baby's brand new immune system looks at the bacterium, sees the sialic acid, and thinks, oh, that's just us, and ignores it. It lets the bacteria hide in plain sight and cross the blood-brain barrier.
That's incredibly devious. So E. coli really does it all. Traveler's diarrhea, HUS, UTIs, meningitis.
Is the versatile prototype. It teaches you everything about how bacteria can acquire new weapons and cause disease.
Okay, let's move to the next family member, the infiltrator, Salmonella.
Ah, Salmonella. First, the nomenclature is a complete mess. Taxonomically, there are really only 2 species, but clinically we use the serotypes as if they were species names.
So we say Salmonella typhi and Salmonella typhimurium.
Exactly, even though technically they're both Salmonella enterica. Now, Salmonella is different from Shigella in a few key ways. First, Salmonella is motile. It has H antigens. It swims.
And it produces hydrogen sulfide, H2S.
Right. That's what makes the black colonies on Hecto and agar. The agar has an iron source, and the H2S gas reacts with it to form iron sulfide, which is a black precipitate.
And the third big difference?
It's acid labile.
Labile, so sensitive to acid.
Which means stomach acid kills it pretty well. To get sick, you need a huge infectious dose like hundreds of thousands or even 1 million bacteria to survive the acid gauntlet and reach your gut.
Unless you're taking antacids.
Exactly. People on proton pump inhibitors have a much lower infectious dose. They're much more susceptible.
Murray calls Salmonella the Trojan horse. How does it get in?
It targets the M cells. These are microfold cells that sit over the Peyer's patches in your gut. They're the surveillance cells of the gut immune system.
And Salmonella tricks them.
It uses its type 3 secretion system to trigger what's called membrane ruffling. It injects proteins that make the M cell reach out and engulf the bacterium. But here's the key. Unlike Shigella, Salmonella stays inside the phagocytic vacuole.
It likes living in the bubble.
It modifies the bubble. It prevents the lysosome from fusing with it, so it replicates safely inside the vacuole, protected from the immune system.
Okay, let's talk clinical presentations. There are basically 3 buckets for Salmonella. Bucket 1, gastroenteritis.
This is your non-typhoidal Salmonella, S. enteritidis, S. typhimurium. This is a zoonosis. You get this from poultry, eggs.
And reptiles, the pet turtle.
The classic pet turtle or iguana. Kids handle the reptile, don't wash their hands, put their fingers in their mouth, and get sick.
And this causes an inflammatory diarrhea.
Yes. Unlike E-tech, the stool will have neutrophils in it. The bacteria invade the mucosa, but it usually stays local. It's self-limiting in healthy people.
And do we treat this with antibiotics?
Generally, no. Studies have shown that giving antibiotics for a simple Salmonella gastroenteritis actually prolongs the carrier state.
It makes you shed the bacteria for longer.
Correct. So unless the patient is immunocompromised, very young, or septic, you just hydrate them and let it run its course.
Okay. Bucket 2. Enteric fever. Typhoid.
This is a completely different beast. This is Salmonella typhi or paratyphi. And this is a human-only pathogen. No turtles involved.
So this is transmitted person to person?
Fecal-oral, yes. The mechanism is different, too. It invades the M cells and gets eaten by macrophages.
But then the macrophages don't kill it.
The macrophages become taxis. They carry the bacteria into the lymphatic system and then into the bloodstream.
So it goes systemic?
It goes everywhere. Liver, spleen, bone marrow. The bacteria replicates inside the macrophages of the reticuloendothelial system.
And what are the symptoms?
You get a stepwise fever. It goes up a little more each day. Headache is very common. And these faint pink spots on the trunk called rose spots.
And what about the diarrhea?
That actually comes later. Initially, you might be constipated. The diarrhea, which is famously described as pea soup diarrhea, happens in the 2nd or 3rd week.
And this is when the bacteria reinvade the gut from their hiding place.
From the gallbladder, yes. Salmonella typhi loves the gallbladder. It can form a biofilm on gallstones.
This is the typhoid Mary scenario.
Exactly. Mary Mellon was an asymptomatic carrier. She had Salmonella living happily in her gallbladder, and she was shedding it into the food she prepared as a cook. She infected dozens of people.
And for typhoid, we absolutely treat with antibiotics.
Oh, yes. This is a life-threatening systemic infection. You need fluoroquinolones or ceftriaxone, and there are vaccines for typhoid targeting that Vi capsule we mentioned.
Okay, last bucket. Bucket 3, osteomyelitis.
This is a classic board exam association. If a patient with sickle cell disease gets osteomyelitis, the bug you have to think about is
Salmonella.
Now, Staph aureus is still the most common cause of osteomyelitis overall, even in sickle cell patients. But Salmonella is uniquely prevalent in this population.
Why is that?
A couple of reasons. The microinfarcts in the gut from sickling might allow the bacteria to translocate into the blood more easily. But the big reason is functional asplenia.
Their spleen doesn't work well.
Right. And the spleen is crucial for clearing encapsulated organisms from the blood. So for Salmonella, it's a direct highway from the gut to the bone.
Okay. Salmonella. Motile, H2S positive, black colonies, acid labile, hides in the gallbladder, and loves sickle cell bones.
You've got it.
Now let's pivot to its evil twin, Shigella, the dysentery specialist.
Genetically, Shigella is basically E. coli that lost its flagella and gained a really bad attitude. DNA hybridization shows they're the same species, but clinically we keep them separate.
So it's non-motile, no H antigen.
Correct. And unlike Salmonella, it is acid stable.
The stomach acid doesn't kill it.
Nope. Which means the infectious dose is incredibly low. Fewer than 100 bacteria can make you sick, theoretically as few as 10.
So it's super contagious.
Unbelievably contagious. This is why we have the 4 F's of transmission.
Fingers, flies, food, feces.
It just sweeps through daycares, nursing homes, and military barracks.
Murray lists the 4 groups.
Right. Group A is S. dysenteriae. That's the worst one. Causes epidemics, produces Shiga toxin.
B is flexneri.
Most common in developing countries. C is boydii, which is rare. And group D is S. sunnae.
Sunnae is sunny.
Right. It's the most common one in the U.S. and industrialized countries, and it's usually milder.
Let's talk pathogenesis. Shigella has this really cool, really destructive trick for moving around.
It invades the M cell, just like Salmonella,
Yeah.
but then it escapes the phagosome. It uses a hemolysin to break out of the bubble and get free into the cytoplasm.
But it has no flagella, so how does it move?
It hijacks the host's own machinery. It has a protein on one end called ISA that triggers the polymerization of host actin filaments. It builds its own actin tail.
It makes a rocket booster out of your own cells.
Exactly. The actin tail pushes the bacterium forward. It rockets through the cytoplasm, slams into the side of the cell, punches through the membrane, and invades the neighboring cell.
So it never has to go back outside into the extracellular space.
Which means it's hidden from antibodies, hidden from neutrophils. It just spreads laterally, cell to cell, destroying the colonic mucosa as it goes.
And that destruction is what causes dysentery. Define that for us. It's not just diarrhea.
No, dysentery is frequent small volume stools that are mostly blood, mucus, and pus. It reflects the destruction of the lining, and it comes with tenesmus.
That's the painful straining.
The painful cramping feeling that you have to go, but nothing comes out because your colon is just a raw, inflamed, spasming tube.
And we have to mention the Shiga toxin again.
S. dysentery type 1 produces the original Shiga toxin. Same mechanism as the E. coli one cleaves the 28S rRNA. And it can cause HUS, just like EHEC. The other Shigella species generally don't make the toxin.
And for diagnosis, on hecto-agar.
It's green colonies. No black center because it doesn't make H2S. That's the key lab distinction from Salmonella.
Okay. We've covered the gut residents. Now let's talk about the zoonotic threat. The plague bringer, Yersinia.
Yersinia pestis. The Black Death. The organism that killed about a third of Europe in the 14th century.
It feels ancient, but it's still around.
Oh, yes. It's a zoonosis. It lives in rodents, rats, prairie dogs, ground squirrels. In the U.S., it's endemic in the Southwest, the Four Corners region. And the vector is the flea.
The flea biology here is incredibly morbid.
It is. The bacteria form a biofilm in the flea's foregut, which blocks the flea's digestive tract. The flea becomes ravenously hungry because blood can't get to its stomach.
So it just keeps biting.
It goes into a feeding frenzy. It jumps from host to host, biting repeatedly, and with each bite, it regurgitates a slurry of bacteria into the wound because it can't swallow.
The bacteria effectively weaponize the flea by starving it.
It's brutal. Once in the human, Yersinia pestis is a machine. It has a special F1 capsule, which is antiphagocytic, and it has something called plaprotease.
What does that do?
It degrades fibrin clots. This prevents the body from walling off the infection. It allows the bacteria to spread with shocking speed from the flea bite into the lymphatics.
And when it hits the lymph nodes?
They swell up, become hemorrhagic, turn black, and are incredibly painful. These are the buboes, hence bubonic plague.
But there's a worse form, right? Pneumonic plague.
Yes. If the bacteria spread from the blood to the lungs, or if you inhale aerosols from someone who already has it, you get pneumonic plague.
And this is contagious person to person.
Highly contagious. And it's almost 100% fatal without very rapid treatment.
On a slide, what does it look like?
It has a classic safety pin appearance. It's a bipolar staining rod. The ends stain darker than the middle.
Okay, there's another Yersinia we need to talk about, the one that mimics appendicitis.
Yersinia enteroclitica.
This one comes from contaminated milk or pork.
And pet feces. Puppies are a major reservoir.
Puppies.
Yep. If a kid gets a new puppy and then a week later has diarrhea and stomach pain, you have to think about Yersinia.
And the unique thing about this one is that it loves the cold.
It grows at 4 degrees Celsius. It can multiply in the refrigerator. This is called cold enrichment.
And that's why it can contaminate refrigerated blood products.
Exactly. It can grow in blood bags in the fridge, leading to transfusion-related sepsis, which has a very high mortality rate.
And clinically, it's the great mimic.
It causes mesenteric adenitis inflammation of the lymph nodes around the gut. This presents as right lower quadrant pain, fever, and leukocytosis. It looks exactly like appendicitis.
So the kid gets taken to surgery.
And the surgeon opens them up, and the appendix is perfectly normal. but the lymph nodes are huge and inflamed. It's called pseudoappendicitis.
Okay, we have done the heavy hitters. Let's finish with the opportunists, the hospital gang.
These are the bugs that hang out in the background until a patient's defenses are down. They are the kings of nosocomial infections.
Let's start with Klebsiella.
Klebsiella pneumonia. The defining feature here is its massive polysaccharide capsule. It's so thick that on an agar plate, the colonies look wet, runny, and mucoid.
Sticky.
Very sticky. If you touch it with a loop, it strings out. And in the lungs, this translates to the classic current jelly sputum. It's thick, bloody, necrotic sputum.
And who gets this?
We teach the 3 A's: alcoholics, abscesses, and aspiration. Alcoholics are prone to aspiration, and their immune function is poor.
And Klebsiella causes a really destructive pneumonia.
Very destructive. It causes cavitary lesions and abscesses. And we have to talk about resistance.
KPC.
Klebsiella pneumonia, carbapenemase. These bacteria have enzymes that chew up our strongest antibiotics, carbapenems. They are an absolute nightmare to treat.
Okay, next up, Proteus. Proteus mirabilis.
The Greek god Proteus could change his shape. This bacterium changes the surface of the agar plate. It has swarming motility.
What does that look like?
It's hypermotile. It doesn't form nice, neat colonies. It sends out waves of bacteria that ripple across the plate. It looks like concentric rings, like a bullseye.
And clinically, it's the stone maker.
It produces a very potent urease enzyme. Urease splits urea into ammonia and CO2.
And ammonia is basic.
So the urine pH shoots up, off or above 8. In that alkaline environment, magnesium, ammonium, and phosphate precipitate out of the urine.
As struvite.
As struvite crystals, these grow very fast and can fill up the entire renal pelvis, creating these massive staghorn calculi.
So if you see a staghorn stone, think Proteus.
Almost certainly Proteus.
Then there's Serratia.
Serratia marcescens. It's famous for producing a red pigment called prodigiosin.
So the colonies are actually red.
Bright red, especially at room temperature. It loves damp environments, so you sometimes see it as a pink film in showers or on hospital respirators.
And it causes hospital outbreaks.
Yes, UTIs, pneumonia, especially in IV drug users.
And finally, Enterobacter and Citrobacter.
These are the cousins. They're both known for being multi-drug resistant. Enterobacter is notorious for inducible resistance with its AMP-beta-lactamase.
Meaning it looks susceptible at first, but then turns on resistance during treatment.
Exactly. And Citrobacter coeseri is a rare but important cause of brain abscesses and meningitis in neonates.
Okay, we have covered a massive amount of ground. Before we get to the questions, let's build that mental flow chart. You're in the lab, you have a gram-negative rod.
Okay, let's visualize the decision tree. Step 1. McConkey agar. Is it a lactose fermenter?
Is it pink?
If it's pink, you're thinking CEKA, E. coli, Klebsiella, Enterobacter. If it's colorless, you're thinking of the bad guys. Salmonella, Shigella, Proteus, Yersinia.
Okay, step 2, differentiate the pinks.
Well, E. coli is indole positive. It breaks down tryptophan.
Yeah.
And it's modal. Klebsiella is indole negative, non-modal, and has those classic mucoid colonies.
Got it. Step 3, differentiate the colorless ones.
Look for H2S production. Do you see black colonies on your hecton or TSI agar?
If it's black.
It's Salmonella or Proteus.
Right.
If there's no black, so green colonies on hecton, you're thinking Shigella or Yersinia.
Step 4, how do you tell Salmonella from Proteus? They're both black.
Proteus has that powerful urease activity and the swarming motility. Salmonella is urease negative and doesn't swarm like that.
And finally, step 5, Shigella versus Yersinia.
Shigella is non-modal, period. Yersinia is modal, but only at room temperature, not at body temp. Now, of course, the clinical history is key. Daycare versus puppies and pseudoappendicitis.
Boom. That is the roadmap.
It's a logical tree. If you can visualize the plates and the key biochemicals, you can navigate almost any clinical vignette.
All right, listeners, it is time to test yourself. We have 5 single best answer questions. I'll read the scenario, we'll pause for a moment, and then we'll break it down.
Let's do it.
Question 1. A 4-year-old child presents with bloody diarrhea and oliguria. That means low urine output. Labs show low platelets and anemia with fragmented red blood cells. The mother reports the child ate a hamburger at a cookout 3 days ago. Which virulence factor is responsible for this clinical picture?
Okay, take a moment on that one.
All the pieces are there.
They are. The answer is Shiga-like toxin, also called verotoxin.
Break it down.
This is the classic triad of hemolytic uremic syndrome, or HUS anemia, thrombocytopenia, and renal failure. The hamburger and bloody diarrhea scream Stech E. coli 0157. Each 7. The mechanism is that toxin cleaving the 28S rRNA, killing endothelial cells in the kidney. And remember, no antibiotics.
Got it. Question 2. A 22-year-old patient with sickle cell disease presents with severe bone pain and fever. MRI confirms osteomyelitis. Blood cultures grow a gram-negative rod that is oxidase-negative, lactose non-fermenting, and produces black colonies on hecto and agar. What is the organism?
All right, another classic association here.
What's the answer?
The answer is Salmonella.
And the clues?
The biggest clue is sickle cell disease, which creates that unique predisposition to Salmonella osteomyelitis. The lab findings confirm it. Lactose non-fermenter that produces H2S, giving you those black colonies. That H2S production is what differentiates it from Shigella.
Perfect. Question 3. A 25-year-old medical student develops fever and right lower quadrant pain. He is taken to surgery for suspected appendicitis, but the appendix looks normal. However, the mesenteric lymph nodes are severely inflamed. He mentions he recently adopted a puppy. What is the likely pathogen?
The poor medical student.
Happens to the best of us. What's the bug?
The likely pathogen is Yersinia enterocolitica. This is the classic presentation of pseudoappendicitis. The right lower quadrant pain from the inflamed mesenteric lymph nodes mimics appendicitis perfectly, and the puppy is the classic zoonotic reservoir.
Got it. Question 4. A 55-year-old homeless man with a history of chronic alcoholism is admitted with severe pneumonia. He is coughing up thick, bloody sputum that looks like currant jelly. A chest x-ray shows a cavitary lesion in the right upper lobe. Which organism is most likely?
A lot of buzzwords in that one.
They are definitely there for a reason.
The answer is Klebsiella pneumonia. You have the 3A's risk factor, an alcoholic patient prone to aspiration. The currant jelly sputum is the key phrase for the thick mucoid capsule of Klebsiella, and it classically causes these heavy, necrotizing pneumonias with abscess formation, especially in the upper lobes.
Excellent. Last one. Question 5. A patient with a long-term indwelling urinary catheter develops a UTI. Urinalysis shows an alkaline pH of 8.5. Microscopy reveals large crystals shaped like coffin lids. The culture shows a gram-negative rod that swarms across the entire blood agar plate.
Okay, this one is all about the lab findings.
What is it?
The organism is Proteus mirabilis. The swarming motility is pathognomonic. The alkaline urine is from its powerful urease enzyme splitting urea into ammonia, and the coffin lid crystals are the struvite stones that form in that alkaline urine.
How did you do? If you missed any, that's what the Murray chapter is for. Go back and review those sections.
These are the high-yield associations. If you know these patterns, you can navigate 90% of the exam questions on this topic.
So we've come full circle. We started by calling them enteric architects.
And they are. They're a family that has truly mastered the art of adaptation. When you think about the host pathogen chess match here, it's incredible.
How so?
The gut is this crowded, competitive ecosystem. E. coli starts as a harmless neighbor, but then it picks up a plasmid, a new weapon, and suddenly it can climb into your bladder or dissolve your kidneys.
It's constantly evolving.
Constantly. Salmonella learned to hide in your gallbladder to evade your immune system. Shigella learned to use your own skeleton to rocket between your cells.
It's a reminder that these bacteria aren't static things in a textbook. They are living, adapting, and constantly trading secrets and picking up new tools.
And that's why the Enterobacteriaceae are, and will continue to be, the most clinically significant family of bacteria.
Ooh.
Because they are always watching, always adapting, and always waiting for an opening.
Whether that opening is a breakdown in hygiene, a compromised immune system, or just an undercooked hamburger.
They are ready to make their move.
A sobering thought to end on. Wash your hands, cook your burgers, and beware the pet turtle. Thanks for listening to this deep dive.
See you next time.